Hello, everyone. My name is Liam Alderson. I'm an honors biochemistry student here at the University of Arkansas in Fayetteville. I work in the research lab under Dr. Kumar, the Department of Chemistry and Biochemistry in the Fulbright College of Arts and Sciences. And I'm honored to be presenting my research at the STEM Posters at the Capitol event this year. So my research project focuses on a specific protein that is of great interest to the Kumar lab, which is human acidic fibroblast growth factor protein. Now, my research project in particular focuses on a certain mutant of this, which is a cysteine-free hyperstable version of it, but I'll get into what the wild type protein does itself and why this mutation is relevant to the research. Now, wild type FGF1 is a mitogenic signaling protein, which means that it interacts with its receptors on cell membrane surfaces. And as a mitogenic protein, it plays crucial roles in bringing about cell proliferation as well as cell differentiation. Some of the physiological processes that this protein plays a crucial role in are embryo development, which is where its proliferation and differentiation roles take place, as well as angiogenesis, which is the formation of blood vessels in cancerous tumors. And of our particular interest in the Kumar lab is the therapeutic potential that this protein has for wound healing applications, such as the healing of scar tissue. Now, one of the negative factors about wild type fibroblast growth factor is that it has a very low thermostability as well as susceptibility to proteases and degradation with that. And so efforts to create a sustainable medical application of this protein would be very difficult due to the thermostability. And you can think about transporting this protein across the nation when you are distributing it, if it is in some sort of lotion, cream, whatever, for some sort of application like that, it would degrade and therefore the medicine would become useless and it would no longer serve its purpose. So my research project proposes a certain mutation that we believe will actually raise the thermostability and lower its susceptibility to degradation, which is a cysteine-free hyperstable variant of human fibroblast growth factor. This mutation, as you can see on the primary sequence, there are 3 cysteine residues in wild type fibroblast growth factor that are at amino acid locations 16, 83, and 117. Now, we are replacing these with serine because we think those are less allosterically inhibiting because cysteine is a fairly large amino acid. One of the reasons we theorize that this mutation will create a more stable protein is because cysteine residues have reactive thiol groups due to their sulfur atoms. And we believe whenever this protein is degraded, it is inhibited from refolding into its biological shape due to these cysteine thiol groups forming allosterically inhibiting disulfide bonds. And so we theorize if we take out these cysteine groups and replace them with serine, this will actually lower this potential and also raise its stability. As you can see, this is a very young research project. I just received funding for it back in January, and so I'm currently in the process of this project. But we've already taken steps, as you can see, with purification of the wild type FGF that will be used to compare. We purified it through Kumar Labs means on where we overexpressed it in an E. coli culture and then purified the protein through affinity binding heparin and cephros columns. And as you can see, we ran an SDS page of the result. And in lane 7, you can see a very intense band, which is actually our purified protein. And the band next to it in lane 8 is actually our wild type FGF marker. So we can see from the SDS page that we were very successful in not only overexpressing the protein, but we also overexpressed the correct protein. Some of the research goals from here on are going to be overexpressing and purifying the cysteine-free mutant of this, and then characterizing this through its structure and its stability, thermostability, and comparing that to the wild type FGF sample that we have already purified. And so some of the future plans for this, we're going to be performing circular dichroism on the cysteine-free variant to check for any secondary structure motif changes to make sure that the protein still retains its functional shape. and we're going to be performing trypsin digestion on the protein itself, as well as the wild type, to compare how susceptible they are to digestion within biological vitro, as well as we're going to be testing their thermostability. We're going to be measuring this with fluorescent spectroscopy to monitor the proteins as they're denatured through various ranges of temperatures, as well as we're going to be checking the heparin binding affinity, because heparin is one of the important molecules that FGF binds to for it to actually carry out its physiological roles. And so this research is going to be continuing through the year 2021, which is my funding period for the SURF grant. And I'm very excited to continue this research project, and I'm very excited to be presenting this, and I hope you enjoy the rest of the event. And thank you for letting me share.